Well, we're very pleased to be talking today with Ronal Pacheco about his recent lab protocol article published in PLOS ONE entitled "A Comprehensive Improved Protocol for Generating Common Bean Transgenic Hairy Roots Under Use in Reverse Genetics Studies," and the associated protocol in protocols.io. Ronal Pacheco graduated from the University of Havana in Cuba in 2013 with a dissertation entitled Pepper Mottle Virus, Biological Characterization and Molecular Diagnostic. He did his honors research at the Laboratory of Plant Virology at the National Center for Plant and Animal Health. He then moved to Mexico and obtained a Master's in Science degree from the National Autonomous University of Mexico in 2019. Ronal is now pursuing his PhD, also at the National Autonomous University of Mexico, in the Laboratory of Mutualistic Interactions in Plants, working specifically on characterizing the roles of phospholipases in the mutualistic interactions between common bean and rhizobia using reverse genetic tools. Ronald, thank you so much for being with us.
Hi, Gabriel. Thank you for the invitation. And it's a pleasure for me to talk about the protocol.
So just to kick off, can you briefly explain what this protocol is about, how to use it, and why it is relevant?
Yeah, sure. Well, in general, this protocol is about hairy root generation in common bean. In this sense, it's important to mention that the protocol is just to generate hairy roots in common bean, because hairy roots are also generated in other species of legume. But this protocol is just to generate hairy roots in common bean. And as you told us, the protocol was published. The step-by-step is published in the protocol.io's platform. But we also published a lab protocol in PLOS ONE, and in this paper we discuss also about how to use it. How to use it specifically how to use it for reverse genetic based studies. So how to use it in general is where to use that protocol first, you have to have a bacteria clone. In this case, is Agrobacterium rhizogenes transformed with the desirable vector, and on the other hand, it's necessary to have some common bean seeds to perform infection with the transformed Agrobacterium clone. So it's in general how to use it. There are many important steps during the protocol, and why it is relevant. The thing is that this is an outdated protocol. It is not the original protocol for hairy root generation in common bean. Actually, the original one was published in 2007, many years ago. But the thing is that over the years, the original protocol has had many modifications, and none of them was published in the correct moment. So, and around 4 years ago, I had to write the material method for an original protocol. And in that moment, I told to my colleague, "Hey, it is not correct to continue citing the old protocol." And in that moment, I decided to write those modifications. I include them in the material, in the supplementary material of that original paper. And after that, we decided to publish our daily protocol in the platform, the step-by-step in the platform of protocol.ios, and the paper in PLOS ONE. So it is relevant in the sense of that the original protocol for headed root generation is not used today like it is exactly.
And can you elaborate on the nature of those modifications that happened since the original protocol were published? How did those modifications make the protocol a better one, I suppose?
Yes, I remember one of the steps in the original protocol. It is to the infection process in the original protocol. The original protocol indicate to perform the infection after plant are around 2 weeks old. And in this in the Oderi protocol is not like that, because in the Oderi protocol the infection step is in seedlings of 2 days old seedlings, and the improvement in this case is that when the infection occur in older plant common bean plants have to be grew up inside pots with vermiculita. Vermiculita is a substrate, and is a little bit dirty. And now that step is not like that. Now is just on the seedling, 2 old days seedling, and is better in that sense. And also another important improvement was the period in this outdated protocol. The period is reduced. I'm not remember exactly how many days, but actually in the material supplementary material of the lab protocol it is a supplementary file with that comparison, and it is another important improvement. And well, the other important improvement was that in the original one, there is no discussion about how to use it and how to select our bacterium rhizogenous clone for plant infection. And in the Oderi protocol, we include that discussion, how to use it, how to take care in the important step, and how to use it for reverse genetic-based studies.
And your protocol in the platform is beautifully written. It's very detailed, tons of images, videos to make it very, very followable and very reproducible. And you mentioned that there are many important steps. Can you elaborate on what steps are the most critical and those steps that the user has to be very careful with?
Yes. It's a pretty important question. I think the most important, the most difficult step during the process, during the protocol, is the infection step, because during the infection step, our person has different skills, have different ability to perform the infection, because during the infection is produced with a needle or syringe on the seedling tissue. And in that moment, the person who is performing the infection has to keep in mind that the person has to produce enough damage, or damage enough to allow bacterial infection, but not to destroy the tissue, the plant tissue, because if the plant tissue is destroyed, well, after that, that plant won't generate successfully hairy roots. So it is the most difficult step, but there are another one. Well, in our laboratory, we, after plants are infected, we include them, or we put them, inside tubes, glass tubes, and around 3 to 5 days. Well, those tubes are has tabs, and after that, it's important to release tabs because if tabs are not released, the foliar area, the leaves of plant don't develop successfully. Of course, it affects the plant in general, the development of the plant in general. Mm-hmm.
And so you early on during this conversation, you explained why you decided to create a step-by-step protocol. And it is my understanding that your motivation was to be precise, to say, it is not correct to cite a paper that is over 15 years old. We are not doing what that paper actually reported. There are many small modifications that have accrued over time, and now we're working with a completely different protocol, and we want to be precise, we want to be accurate, and we want the reader to be able to reproduce what we're doing. However, you took that protocol and sent it to peer review. What was the motivation to have the protocol peer-reviewed in PLOS ONE?
Yes, well, in this sense, I think I had two main motivations. The first one is the possibility to publish the protocol on the platform, the protocol.io platform, because the original one, the old protocol, was published in a different way, and I think publishing the protocol, the step-by-step on the platform, is pretty, it's beautiful. The platform is beautiful because researchers have the possibility to include video, photos, etc. And those things make the protocol more understandable. And on the other hand, another motivation, well, in this case, to publish as a lab protocol in PLOS ONE, well, I think, well, I knew a few years ago, I knew that there is an agreement between protocol.io and PLOS ONE, and that brings us the possibility to make publish a step-by-step protocol like a lab protocol. So in that moment, it was a great an important motivation, because I remember in the moment I told to my boss, hey, I think this is the right moment to become the protocol published for all the researchers in the area. And of course, in this sense, it's important the things that PLOS ONE is an open access journal. So I think it was a great opportunity to publish the protocol on the platform with all the details, with video, photo, etc., and to make it published like a protocol, like a paper on PLOS ONE. It was pretty important, and it was there were my main motivations.
And how did the feedback that you received from the peer reviewers shaped the final version of the protocol?
Well, it is a funny question, because I remember during the process, the publishing process, well, it is the first time that I participate in writing that type of paper. And I remember in the first version, I did include some information in the material method session. And one of the reviewers asked for that information. And it was pretty important to improve the final version of the protocol, because after that I include that information, and of course I realized that it was important to improve the protocol. So the peer review process is essential for the quality of the paper.
And you decided to make those comments from the reviewers public. Yeah. And available in PLOS ONE together with the manuscript. Can you tell us why?
Yes, my opinion about science is that science has to be as transparent as possible. So any data, any information that could be published for other the researcher in the area is important to make it published. And yeah, also, I think if another reviewer or another researcher who are in the publishing process have the access to that information, can be better, because that information can help to a better understanding between them, to better interchange between them. And I think it's a great idea of, in this case, PLOS ONE, to publish the history peer review.
And the last question that I have is, this is a very young paper. It was published actually less than 1 month ago. So we might be speculating at this moment, but what do you think, or what has been the impact of this peer-reviewed paper and the protocol for your career, or what do you think is gonna be?
Well, as I told you before, it is the first time that I write that type of paper, and it was a challenge for me. It was a challenge because in any moment, in some moments, I felt a little bit lost how to write that type of protocol, because it's not similar to another type of paper. So, and that's why I didn't include some information, for example, in the material method section. And it was challenge for me, and for me that as a young researcher, it was a great experience, because now I know how to do it first, and second, I know about protocol RIOS platform. I know about the agreement with PLOS ONE. Now I'm working and I'm thinking in a new protocol that maybe I can produce later. So it was a challenge for me, and it was a great experience.
And I don't have any more questions, but if there is anything that you would like to share that we haven't discussed so far.
Yes, actually, look, it was something accidentally. Last year, I was working in another laboratory, and I had a problem with the growth chamber, with the temperature of the growth chamber, and I had to incubate plants after infection. Infected plants have to incubate in a lower temperature. So in our protocol, we suggest to incubate plants after infection at around 28 Celsius degree. And because the problem, I had to incubate plant under around 25 Celsius degree, because it was another type of growth chamber. And after 12 days, which is the normal period after which plant began to generate hairy roots, I realized that almost all plants generate hairy roots. And usually the efficiency that we have is around 33%. And now in that moment, I realized that around 95% of the plants generate hairy roots. So now we are performing the protocol like that, but of course it is not included in the lab protocol because it occurred after the protocol was accepted. But we are thinking about publishing a new version on the platform to include that modification, because it's pretty important. Pretty important because one of the main disadvantage of the protocol is the efficiency of the number of plants that successfully generate hairy roots. But it was accidentally.
Well, Ronald, thank you so much for sharing your knowledge, for discussing this paper, for discussing the protocol. and sharing your take on reproducibility, open access, and peer review.
Okay, thank you. Thank you for the invitation. Bye.